Good evening, everyone. Today we will start with the new topic, luminometry. This luminometry, it comes from the two words, luminous, okay, and then metry. Luminous, it is basically, it is associated with the light, okay, and metry is any device which helps in the measurement. Okay. So till now, we have been discussing about several spectroscopic techniques, wherein we have told that in any of the spectroscopic technique, that electromagnetic radiations, they interact with the object or matter, and based on their interaction we get some output signal, okay, and the measurement of this output signal it provides us information about the concentration, about the molecular architecture, okay, and the structural information. Information about the molecules, okay. So in this regard, we have talked about first, we have talked about UV vis spectroscopy. In the second instance, we have discussed about fluorescence, and phosphorescence. In the third instance, we have talked about this Raman spectroscopy. Okay, and yeah, these were the main things. And anything else we have talked? Visibles, fluorescence, and yeah, Raman spectroscopy, and yeah, mainly these three we have talked, right? Now, in all of this, if you see, we had an electromagnetic radiation and it interacted with the molecules, and we got some information from there, right? Now, this luminometry, it is a specific kind of uh, spectroscopic system wherein we do not need a source. We do not need a source, okay? The source is within the sample itself. Okay. Now, if you look into the fireflies, so what do? They glow at night. Now, this glowing at night, it is nothing but luminescence. In fact, when we talked about this fluorescence and phosphorescence, we also talked about luminescence, and therein we discussed that both fluorescence plus phosphorescence together they are regarded as luminescence. Okay, I hope every one of you remember. Now, if you see this glow at night, the light which is being generated from within the system, they are called cold radiation. Okay. Can anyone tell me why it is called cold radiation? Any guess?
There is
Anyone?
no heat involved.
Because there's no photon involved.
No photon involved. Sorry to say that whenever there is some light which is coming out, if you do not have photon associated with that, then you will not be able to see any radiation, visible radiation. So that is incorrect. And someone told that there is no heat involved. It is partially correct. Uh, it is regarded as cold radiation because the heat which is being generated, it is not sufficient to alter the biological processes within the fireflies. Okay, because if there is a significant increase in the temperature within the biological system, they would denature all the bioactive compounds or bioactive polymers, and if that happens, it will make the system dead. Okay, so the radiations which are being generated over here, they are so weak that they do not increase the temperature. This sign, this C cap, it is a sign of width. Okay. I hope many of you know. So, so the cold radiation, it is regarded as such because the intensity of the radiation is so weak that it does not increase the temperature within the biological system. And hence it does not interfere with the biological activity. So the biological activity, it is not being hampered. Okay, so due to this reason, the radiations which are coming out through the process of luminescence, it is regarded as those radiations are regarded as cold radiation. Okay, now if you look into our previous systems, say for example this fluorescence and phosphorescence, what was our architecture? Our architecture was, we were having a source, okay, then we were having some collimating system or lenses and collimating system. Then we were having the object, okay, then at a right angle we were again having some lenses or filters, right, or even we could have this monochromators, whatever. In total you say some optics. Over here also you name it as optics. And then we were having the detector. Now, as we discussed herein, what is happening in the fireflies where the luminescence is being generated? It is being generated from within the fireflies, right? So there is no need of source. So this area, which I am marking with black, that is the source and lens or collimating system. This optics, this the which is getting us a light, which will be making the radiations or the electromagnetic radiation incident onto the sample. That optics part, it is not present. Okay, so we will be in lumiноmetry. Basically, we will be dealing with the sample and then the optics to have some filtering and all this thing, and then we will be having the detector. So all the detectors what we discussed in the case of fluorescence and phosphorescence, they will also hold good over here also, over here. Okay, like we discussed about PMT. Okay, we discussed about imaging based sensors. So all these detectors, they will also be used over here. Okay, now let's move forward. So background, what is a luminometer? As I told you, it is an instrument or a device that measures weak emissions of visible light which are coming from a sample. Okay, now since the lights which are being generated from within the sample, we are analyzing that, hence there would be no source. Okay, usually when we deal with biological samples for our analytical purposes, we will be seeing at a later stage in a few moments of time that the reaction which is going on within the firefly, we use that mechanism and we try to emulate that within the biological system to analyze some of the what you call chemical constituent, or we can also emulate it to have an understanding about the interactions between two proteins and so on. Okay, so when we are usually dealing with these biological samples, say if you want to analyze this blood samples, okay, so we cannot get a large amount of sample. Usually we have to work with a small amount of samples. So hence the luminometry, it usually is being designed to work with small samples. Okay, and sometimes it is also required. Suppose if you want to analyze the DNA and all these things, then in those cases you may have only a few microliters of the samples to work with. Okay, and as I already told you that samples emit light by themselves, right? And due to this reason, we do not need an excitation light. We do not need an excitation light. This is a very, very important thing. So if I give you a short note to describe between the UV visible spectroscopy and the luminometry, this should be one of your important points. Okay, that there is no excitation light. Okay, so because of this reason, there are no lamp or the excitation optics. Okay, and luminometer, conventionally they use photomultiplier tube, which I have already discussed with you, PMTs. Okay, and these PMTs, another important thing, these PMTs, they are placed very close to the sample. Why they are placed very close to the sample as compared to the UV vis or fluorescence? or phosphorescence spectrometer? This is because in these type of spectrophotometers, we have an excitation light and we are analyzing that excitation light. Okay. However, in the case of luminometer, what is happening? Whatever the light are being generated, they are being generated within the biological system or within the reaction system which we have put into the microplates, right? So the light what we will be getting, they are weak emissions, right? In the first point, whatever we have discussed, these are weak emissions of visible light. If we place the PMT at a greater distance, suppose here is the sample, and these samples, it is emitting light in all directions, right? Now, if you place the sample, this PMT far apart over here, if you see a large amount of the radiation, it is being lost. However, if you bring this PMT near to the sample surface, this cuvette, then it would be able to capture a greater number of photons. Okay. This is why when we are working with luminometers, these luminometers have the detectors which have been placed very close to the photon. Nowadays, with the advancements in the solid state electronics, you need not to put the PMTs just near to the cuvette or the sample holder. Instead, we can have fiber optics around this sample holder. So you can add a large number of the fiber optics, an array of fiber optics, which can collect all the information from the fiber optics, and then they will deliver the photons to the photomultiplier tube. Okay, so in that way, you will be able to get a large number of photons, a greater amount of photons into the PMT, and your signal to noise ratio will increase. However, even though we have made several advancements in the field of electronics, but we do not have control over the physical processes. So, in the case of microplate luminometers, the PMT needs to be kept away from the samples to prevent crosstalk. Now, what is a crosstalk? Can anyone tell me? What is crosstalk? Anyone? You can shed some information on crosstalk. The meaning is within this word itself, crosstalk. What is cross? So this crosstalk, it is made up of two words, cross and talk, right? So over here, say for example, you are here, you want to talk with your person C, a person B. So you are A, you have your friend B. Okay, so you picked up your mobile and you have dialed him or her, the person B. However, due to some network failure, you connect to person C and not with B. It many a times it happens with the phone connection, and at the previous stages or earlier days, it was more prevalent. Okay, so this is nothing but it is called crosstalk. So you intended to talk with the person B, but unintentionally you are getting connected with person C. Okay, so this is crosstalk. So that means unintentional interference or interaction with another person. Okay, so crosstalk is an unintentional process. Are you getting my point? Now, what is a crosstalk? As we discussed, it is more prevalent with microplate readers or microplate readers. As I told you over here in the previous case, over here we have an opportunity to what do you call to interact with or to analyze the samples which is having a very small volume, which is in the range of or in the volumes of microliters. Microliter is very small. Now, how do you handle this kind of samples? To handle this kind of samples, we have microplates. You can write the full name as microplates. But in many books, in a short, you can write it in this way. This is micro sign, a hyphen plate. So what is a microplate? Microplate is nothing but a rectangular polystyrene casing, and within that you have an array of wells. And these wells, they are very commonly used in biological purposes or biological analysis. Have you gone through the course tissue engineering?
This is in this current semester.
Okay. So there has been a lot of time. Have you discussed about how the these cells are being analyzed? Mammalian cells are being analyzed, whether they have proliferated or not?
Not yet, sir.
Yes or no? Not yet. Okay, you will be studying over there. So how it is done? Suppose you want to measure the cytotoxicity. Cytotoxicity of a number of samples. So what you do? So you have these microplates, and within this microplates, you can put in your samples. You can seed your cells, and then you do some analysis on or you put some these reagents which will interact with your biological cells and all these things and give you some coloration. And that color, whatever the color it is being generated, it is being read by the microplate reader. Okay. So these microplates, it allows you to handle a number of samples at a time. Okay. So it gives you that flexibility. One of the common this microplate wells, it is in or a microplate is a 96 well plate. Okay. Now they are made up of polystyrene, high grade polystyrene. Now, polystyrene, it is transparent in nature. Right. Now suppose you want to analyze the sample in this central pixel or central well. Okay. So this is a schematic representation of the microplate. Okay. Now in this at this central well, you want to analyze the luminosity. But what? Can you control in which direction? Suppose you have the well. Can you control in which direction you will be? You intend to take the what do you call sample signal? You can have a detector at a particular location, but this sample, it is radiating the lights in all the direction. Now, when it is radiating all lights in all the directions, over here, what it is doing? It is contributing some of the photons from here to here, here to here, here to here, and so on. Right? So, when you will be analyzing these other samples, you will be having interference from the what you call from the well, which is from the adjacent well. Okay, whatever the wells are there. Okay, so it is not just from here to here, it is also in a reciprocative manner also. These other wells, they are also contributing some photons to the central well, right? So there are chances a signal produced in a well of a microplate, it is affecting the signals of the adjacent wells. Okay, so this is called crosstalk. Okay, any question till now? No.
Sir, the same
Okay.
question.
So
that
instrumentation.
mentioned- Excuse me, sir.
So
the question is where to place the detector then?
Where to place the detector? That's what I told you. This is a phenomena which you cannot prevent. It is a physical process. So you need to place the detector as close to the wells as possible. Because if you look into the previous slide, so if you place it more closely to the sample cuvette, the radiations from the other cuvette, it will be very difficult for them to gain entry into the detector. But having this kind of stuff, you cannot stop this information. Okay, to avoid these systems in many microplate readers where you want to do the luminescence analysis, what happens is that these wells, they are sometimes these wells, if you look, they appear like this, right? So you can place the detector over here. And then what happens is this, the walls of these wells, they're coated white. Okay, so they are made opaque, and at the same time they are reflective in nature. Now, due to this reflection, what is happening? This sample where these photons will go, ultimately these will go and they will get what you call they will be coming out through the bottom of the samples from the well itself, right? And due to this reason, the loss of photon will be quite low. Okay, and you will be able to get a greater or higher signal to noise ratio. Okay. Usually, the normal microplate readers, they are transparent. But in the case of luminometer, you can this code these borders of the wells, as white, so that first of all the radiation, it cannot go out of the way and then you can collect the information only from the bottom of the wells. And secondly, if you can say that I will paint it with black. Okay, if you paint it with black, the black coating, it will absorb some photons, right? Am I clear?
Sir, but the
So
last line
the
was
coating the wall. Please.
sir, the last line was we
Please.
should keep it away from the sample. Detector should be kept away from the sample in case of microplates.
No, 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 no. If you see, initially I told you got it wrong. Initially, you had this detector over here, and then you placed it close by. Okay. However, with the advancements in this technology, we have the fiber optics. Fiber optics cables are there, so which can collect. You can place an array of fiber optics just close to the this cuvette, and you can take this fiber optics away and keep it at an what do you call? What does the fiber optics do? Fiber optics, it collects the light from the sample, right? And then it can carry at a distant place where the PMT has been kept. Okay, but the collection of the light over here, if you see the collection of light, it is from as near as possible from the cuvette. Am I clear now? Hello? Am I audible?
Yes, sir. Yes, sir.
Okay. So the thing is that if you keep the detectors away from the cuvette, that means you will be losing signals. If you see, you will be losing signal. You have to keep the detectors nearby. However, nearby means so that your detector can collect as much photons as possible. However, with the advancements in the solid state electronics and this fiber optic communication, you can use fiber optics to collect the photons from as close as possible from the cuvettes, and you can transport those photons to a greater distance where the PMT has been kept. So this gives you a flexibility. But here also the collection of the light photons should be done from as near as possible from the cuvette. Okay. Now coming to the instrumentation part, I've already told you a basic of this one. So what is happening? You have the cuvette over here. Now you cannot do it in an open area because the other room light and all this thing, they will interfere with your measurement. So you have to keep your cuvette within a light tight chamber. Then you collect the information, you get the photo multiplier, you have the amplifier, and then you have the detectors over there, and you detect the signal. Okay, so this is a simple diagrammatic representation of a luminometer. So these are the basic components. Okay, however, the thing is that if you look over here in the second picture, you have this PMT, and you have the from here you have the sample, then you have a monochromator or a filter. What does this monochromator do? It helps in spectrum. Okay, spectral decomposition में क्या करेगा? You are having a light, you have a monochromator, then Vibgyor. In the visible spectrum, it will decompose the light. Okay, and then that can be recorded either with the PMT or with the image-based systems, imaging systems. Okay, so that you can get a full spectral view. In normal luminometer, what you will get, you can only get some this readings in the relative light units. How many photons are striking over the your sensor? Okay. Whereas when you use imaging-based sensor or a monochromator, which can decompose the spectral composition, and then at several spectrums you can get this information that at this wavelength this is the reading, at this wavelength this is the reading. So across that you can get the spectral reading. So the spectral reading would be something like this, lambda, and in the y-axis you will be having intensity, and the intensity is in RLU, relative light units, and spectral band like this. Okay. Or if you want to go for this relative light for this cheaper ones, so the cheaper ones, they will not decompose this light. It will just be having this automultiplier, amplifier, and detector. So you will not have any wavelength specific information. No wavelength specific information will be obtained. You will just be getting information about the intensity. But this intensity is coming from which wavelength? You will not have any information about that. Okay. Any questions over here? No? Okay. Now, other important parts of a luminometer, the dark chamber, as I already told you, that it is needed because it is the area where samples are measured, has to be absolutely protected from the external lights. If external light, they gain entry into the sample, they will interfere. Okay. Then injectors. It is a very important thing. Suppose you are doing some kinetics experiment, chemical reaction. Say, for example, you are doing some chemical reaction. So you have this cuvette. Okay. In this cuvette, you have a particular, say, solvent or something, say A, or a solution of A. Okay. Then you have this injectors. Through this injector, you are infusing B over here into the solution A. So when this is happening, this A and B, it reacts to form C, which will give you luminescence. But it is a very fast reaction and you want to catch it at what instance you are getting the information. So what you can do is that you can have this computer control system where you will be having the time. Okay. Then in the y-axis, you may be having relative light units, that is the intensity in relative light units. Okay. Suppose you start the instrument, say at this T0, you are injecting the sample, it is automatically documented, and you get an into intensity something like this, and maybe it dies down over here. So at say this point where there is an increase in the luminosity, you can say that it is the initiation of the formation of C, and it reaches to a point, highest point at say T2. So this is T2, where the reaction is complete and the sample, this reaction is say it stays constant, and the production of C is being completed at T2, and up to T3 it stays over there, it is constant. That means neither there is a production of C nor there is a destruction of C. However, after T3, if you see, there is a reduction in the intensity level, and that reduction in the intensity level, it indicates or suggests that the C component, it is getting destroyed. Okay. So in this way, you will be able to understand. So say, how do you try to find out the kinetics? So at what rate this luminosity has increased? So you will be able to tell at what rate the C has been produced. So you will be able to do some kinetics study. Also, if the C component it is getting destroyed, you will be able to, sorry, you will be able to analyze the decomposition kinetics. Okay, so you can try to find out the synthesis kinetics or the decomposition kinetics, whatever you want. And many cases, what happens? This information, it is there only for a few seconds. So the injectors, they help us in getting this information. Okay, because suppose if you try to do it manually, so when will be your T0? So you're having a couple of seconds. So by the time you will be adding, you will be closing already 5 to 10 seconds would be gone. Closing means closing the door of the this dark chamber and all this thing and setting up everything. By the time you have completed all these steps, this kinetics information, it might have been lost. Okay. So these injectors, it plays an important role, and these injectors, they are regarded as the reagent injectors. Okay. Any question? No? Okay. Now types of illuminator, tube luminometer. These are very affordable. You can also make it in your lab. Okay. If anyone of you are interested, we can definitely develop it. So if you are interested and if you come to the lab after this say pandemic gets over in the final year, then we can work with that also, if anyone is interested, obviously. So over there, what you need, you have a test tube. Now you can use this test tube over here, and if you, it can act as a cuvette. You place it in a dark chamber, and whatever the information the information you are getting over here from here, you collect it and you get the information over there, and you will be able to get the intensity. Okay, so you need to put some detectors inside that, and once you have the detectors inside this black chamber, you will be able to capture the light over there. And it's very simple. And nowadays this this analog to digital converters, you will be having these detectors. They are nothing but analog sensors. So you can have the analog to digital converters, and through that you can take it into the microcomputer or somewhat in those things, and you can calculate the concentration, and you can devise it at a very cheap price. Even you can devise it in your own lab. Okay, because of the ease of construction. However, if you want to design a microplate illuminators, as I told you, within a very small area, you have to do the analysis for a large number of the sample, and there are various techniques or various what you call problems which are associated with that. And one of the major problem is the crosstalk. Designing microplate illuminators within the lab, it would be bit difficult. Okay. So these are usually being carried out, or usually being designed, or they are there in very high-tech labs, where because the cost of these microplate illuminators are very high. You can design this system in your own lab at a price of say rupees 10 to 20K. However, these machines, this microplate illuminator, it would cost something around rupees 5 lakhs, 5 to 10 lakhs. So yeah, some cheaper things are also there, which may cost to around 3 lakhs also. Okay. However, 3 lakhs, it's not a very small amount. It is also a very high amount. So you can design your own system within 10 to 20,000. Okay, and if you want, you can also, if you don't want to have a handy one, you can use your own computer, and anyone can use it to do the analysis. Okay, so my intention is to tell you that this micro plate illuminator, it would need a lot of miniaturized electronics, to develop this illuminators. However, in tube illuminators, you can design it very easily. Okay. Then there are multi-mode microplate readers. This enables different testing modes like ELISA, fluorescence, luminescence, and applications on a single platform. So as I was telling you, the cost of this microplate reader, it it is around it starts from approximately 3 lakhs. This kind of system, the price is approximately more than 10 lakhs. However, if you try to get this ELISA, fluorescence, spectroscope, luminometer, and luminometer, and then this absorbance, UV visible absorbance. Basically, over here, visible absorbance is mainly checked. If you try to get all these things separately, it would cost you somewhere, it would be more than rupees 20 lakhs. However, if you want to get it in a multi-mode system, it would be costing you approximately 10 lakhs. So that is the advantage. Okay, so if you are able to see, if you see the price difference, these tube illuminators, if you try to get it in a commercial level also, it would be approximately 1 lakh. Okay, So 1 lakh to 3 lakhs, where you over here in the first case you are only analyzing only one sample. In the second case, you can analyze multiple samples, like 6 wells are there, 12 wells are there, 96 wells are there. Okay, and higher wells are also there. I can't remember it properly. So these are the most common thing. So if you see the price jump, it is only 3 times. And but however, you are able to get that things. The 96 samples you are able to analyze at one time, and also you are able to handle lower amount of samples, which is not possible in the first case. Okay. Now you see the price jump. It is only 3 times. Now from here to here, it is again around 3 times, approximately 3 times. So the more the complex system you acquire in your lab, relatively the price would be cheaper. Okay. Now today I will stop over here.